In this lecture, I will provide an overview on different aspects of bioinformatics we learned in this course, specifically on various algorithms or databases as well as the different applications of bioinformatics. So, we started with the fundamentals of bioinformatics, right. So, what are the various fields of science which contribute to bioinformatics? Yeah, mainly the biological sciences, right. Biological sciences provide a wealth of experimental data, because data is very important. So, experiments provide a lot of data, put in databases and then sequences, structures, pathways, networks, right. So, various information we get from the biology. So, then information is very important, information technology plays a major role, as well as other disciplines like physics, chemistry and mathematics, statistics, right. We discussed about all the aspects, how the different fields of science contribute to this interdisciplinary area. What are various features of bioinformatics we discussed? Development of databases and algorithms. We can derive some hypotheses, right. For example, if we have the data, right, and we will try how we generate this data and what are the factors which influence, right, to get that specific data. So, we can give some hypothesis, and based on the hypothesis we derive some important features, right, and if you get some properties, then we can relate, right. We can try to develop some models, prediction algorithms, right, and we can use it for the validation. Right. Then also we discussed about the structure based drug design, how the different aspects of this bioinformatics tools and databases are useful, right, to identify some lead compounds for different drug targets. Then we discussed about different applications of bioinformatics, and then how the bioinformatics contribute in different complexities of biological systems. We start with the DNA sequence, and then DNA structure, and then protein sequence, protein structure, protein interactions, and then this interact networks and the pathways, then the organism, the organs, tissue, right, up to the organism. So, various aspects, right, you can see in each category we see the applications of bioinformatics either in the databases or the models or servers, right, and different ways, understanding these complexities of these biological systems. Right, then we discussed each aspects separately. Mainly we focused on the protein side as well as some aspects of DNA. Right. If you discuss about the DNA, what is the difference between DNA and RNA?
2 prime events.
Right, one is H and OH, right, the sugar, and the base. So, what is the base in the DNA? Thymine, and the RNA? Uracil, right. You can see the difference between the DNA and RNA. So, then we discuss the complementary strand, right. We get the this is the AGC, right, if it is 5 prime 3 prime, right. What is the complementary strand in the 5 prime 3 prime? Right, G C T A T, right. First we get this strand, this same 3 prime 5 prime, then we change the inverse, right. So, we change the direction, then we change this sequence, right. Then we discussed about the transcription translation, right, also the degeneracy of this from the codons, right, to the amino acids. And what are software we discussed on the translation as well as this complementary strand? EMBOSS, right. We discussed about EMBOSS. So, what is EMBOSS? European Molecular Biology Open Software Suite, right. So, this contains various packages. So, we can use this package to get the various parameters of this DNA. Right. Then we discussed about this reading frames. What are different reading frames? 3 for the forward and 3 for the backward. So, 6 reading frames. Then what are the various sequence based parameters we discussed? Where we can see the flexibility and the rigidity, right, and the base pair preferences, right, the compositions, various other aspects, right, based on this DNA sequence. Because this DNA sequence is also important, right, to see these parameters to understand the various functions of these DNAs and this, how they interact with these proteins. Then we moved on to the different types of databases. So, when you start with database, what is the database? is a collection of data in the computer readable form, right, because we have several information available in the literature on various aspects, right. So, various biological data, so they publish in literature, it is very important to collect and put in a proper database, right. So, what is the characteristics of databases we discussed? Right, first we discussed about what are the what we need the contents, right. Then we need the ontology. What is ontology? So, because when we develop a database, there are several technical terms that are used in the database. So, we need to give the complete details of this database, right. Then what are the other characteristics? Schema. What is schema? Right, how to put your data, right, what is a major aspect, right, you have this is your main information, then supplemented with other information. For example, what are the direct information related with this main data. For example, if you take the thermodynamic data, we need the protein name and the source and the experimental conditions and on. Then we can give more additional information regarding the sequence, structure and other things, right. Then we give the search option. So, then we have to give the display, right, how to output your data. So, you have to make the organization right of your database, how to do that, right. Then what are the next one? You have the specific format. What is format? Right, they have the same order. So, you have the order, what are the terms you use in your database. So, it should be the same in all the entries, right. Then it will be easier to fetch the data, right, as well as for the users it will be easier. For example, if you take the protein data bank, start with the header, compound, right, finally it goes up into the sequence residue information, then second is the helix strand, then start with atoms, right. So, if you are interested in the 3D structure, 3D coordinates, then you go with the atom records, right. So, if you see all any PDB ID, so they follow the same format. So, you have to use some specific format. And then the data retrieval, right. How to retrieve the data? Like we should provide proper search options. It is to think about what are the various options users like to have, right. Personally, if you use your thermodynamic data, they are interested in the free energy data, right. Likewise, what are the information the users prefer? Accordingly, you design your observer site so that they can easily obtain the data, right. There are some search options. Then as soon as they search, they should get the reliable data, right. It is very important to get this reliable information and the information what they require, right, that is very important. So, in this case you have to make the interface such a way that the users can retrieve the data, right. Second aspect is that should be available all the time, then should be fast enough, right, to show the data, right. So, otherwise if it is not properly maintained or it is not available all the time, then the users do not use it again, right, they have to be very careful. Then we have to provide proper links to the external databases, because when you develop a database you get the information from different resources. On the other hand, there will be several other databases which are related to your specific database. In this case, we have to provide the links to all the relevant databases. In this case, users can get the information, right, if they need more data, right. For example, if you take the stability data, so you get the data from the literature, provide the literature information. In this case, they can expand, right, the knowledge to read the papers. So, like the database, you have several characteristic features, and we discussed about the various databases. What database for the nucleotide sequence databases? Now, you see the DDBJ, EMBL, GenBank, right. So, they have the different information or they have similar information. They have similar information, right? They also follow similar format also. So, the same time they develop different places, then they form the network. So, in this case they can share the data. So, in this you can deposit data in one place, it can share by others. In this case you can get the uniform data in all these databases. Then we discussed about different properties, right. Nucleic acid property database which contains data for the different base pairs, right. For example, all different dinucleotides, for example, AA, AT, AC, AG and so on. like the flexibility or the rigidity and the stability, melting temperature, right, the stacking energy, hydrogen bonds. So, they have all the information in this database. So, you can use this information available in the database to see if you have any sequence, right, what is the average flexibility or average rigidity, because sometimes the the DNA should be flexible enough to interact with the protein. So, this you can see if you use this data from this nucleotide property database. The literature database, right. So, what is the major literature database for the biology? PubMed, right. So, there are various databases like physical abstract, chemical abstracts, biological abstracts are available, but currently for the biologist and the medical field. So, PubMed is very widely used because it includes the data from various literature, for various journals, right. They cover various journals and it is a wide variety. So, you can get sufficient information from the PubMed database. Addition, we can see the Scopus or the Science Citation Index, right, Web of Science, that also you can use as literature databases. So, then we move to the protein side, right. So, what is the building block of proteins? Amino acids, right. So, it can how many different amino acids use? 20, depending upon the characteristic features, how we classify these amino acids? Hydrophilic, hydrophobic, right. You have the hydrophobic and hydrophilic. What are subclasses? Okay, we take the hydrophobic, aliphatic, aromatic and sulfur containing amino acids, right. What are sulfur containing amino acids? Methionine and cysteine. So, we discuss cysteine, right. Cysteine can form these disulfide bonds, right. This is also important for the hydrophobic interactions. With the hydrophilic, it is a polar and charge. The charge we have positive and negative, right. What is the negative charge amino acid? Aspartic acid and glutamic acid, right. Glutamic acid and aspartic acid. Positive charge, lysine argine, right. You can see the different types, because these amino acid residues, they form specific types of interactions, right. So, that they can form the compact globular shape and maintaining the stability of particular protein. So, then we discussed about the protein functions. What are the various functions? Enzymes, cell signaling and antibodies, right, transport proteins, right, switching, right, and the structural aspects, right, blood clotting proteins, antibodies, right. So, you can perform various functions, right. We discussed about various types of proteins, globular protein, membrane protein and the fibrous proteins. Each one of them, they are responsible for different types of functions, right. So, then we want to understand the structure function relationship, right. The function depends mainly on the structures. So, it is necessary to understand about the functions. So, what are the different types of protein structures? Right, primary, secondary, tertiary and quaternary. So, what is the primary structure? This amino acid sequence. So, here the English sequence, you know the covalent bonds, but we do not know about the other location of the amino acids and all. The secondary structure? So, regular arrangement of these amino acid residues, right. This is mainly the alpha helices and the beta strands. Tertiary structure? That will provide the exactly the coordinates, so where the location of all the atoms, right, in the residue as well as in the complete protein. What is the quaternary structure? Different subunits. So, protein has different subunits, they form to have this quaternary structure, right. So, in between there are two different structural aspects, you can see the supersecondary structures plus the domains, right. Then we see the different types of databases for protein sequence, protein structure and protein function. We take the protein sequence database. So, what is the initially developed protein sequence database? PIR, right. They developed the protein information resource and then they developed this SwissProt. So, this PIR and SwissProt, they merged together to form this UniProt. UniProt has the manually curated sequences plus computer annotated sequences. Currently, how many data in the UniProt database? Around 80-85 million sequences, right. And what is the unique features of UniProt? Right, next less redundancy, highly integrated, and the links with other databases, right, the integration of databases. So, this is the reason why the UniProt is widely used. Also, what are the information which are available in UniProt database? You can see the functions, you can see database data, and you can see the pathways and interactions, right. If you have a protein, so you get almost all the information, right, from this UniProt database. So, then how to obtain the data from UniProt? You can search, right, you can search with any protein name, or you can search with the mesh terms, right. Also you can get the data with specific redundancy, right. You can what are various cutoff available from the UniProt, 90%, 100% and 50%, right. You can search and then you can get this information. You can get the data for any specific protein as well as a group of proteins you can use, right. You can use different mesh terms, right. You can obtain the data. Then what are the applications of UniProt?
To a sequence protein.
Yeah, you can take the sequence for any aspects, for whatever the classes are based on structure, based on function, right. If you want to understand the specific features, right, you can get obtained from this UniProt. So, also UniProt provides the statistics of the different proteins. What is the average length of the protein in the for the sequence development database? 314, 15 sequence residues, right. But generally, they are only about 100 to 300 residues, right, and some proteins are long length. So, average you can say about 315 residues. Then also they give the statistics for the different amino acid residues. Which residues are highly occurring amino acid residues? Alanine, valine, leucine, these are having about 10%. Then few residues, the rarely occurring amino acids. What are rarely occurring amino acids? Cysteine, tryptophan, right. This is rarely occurring amino acids in this UniProt database. So, then we have these sequences. They want to compare, right, how far two sequences are similar, right, how they are distant from these sequences. So, we try to understand this sequence similarity, we develop algorithms for the aligning sequences. So, what is pairwise alignment?
Alignment of two sequences.
Align two sequences, right. So, what are the various ways to align two sequences? For example, this is a sequence. Right, you can first you try to use the without gaps, then you can align with the gaps with gap penalties, right. Then we see the alignment based on some scores. How to give scores? Matching score. What is the matching score? If it matches, okay, with this the alignment, for if you put like this is this matching, so this is a match score. So, what is then mismatch score? What is mismatch score? If we take this is not matching, so you have to give a mismatch score. Then what is gap penalty? If there is a gap, then you have to give penalty for this gap, because compared with these mutations, gaps are rarely occurring. In this case, you have to give more penalty to the gaps than the mismatch score. What are different types of gaps we discussed? Which penalties? Origination penalty and the gap penalty. Gap means number of number of gaps. This is how many times gap originates. This is the how many number of gaps. Right? For example, what is origination penalty here?
1
is 1, because gap originates only once. What is the gap penalty?
2.
There is 2, right, because 1, 2 gaps, right, like that. Then we try to discuss about the scoring matrices. What is scoring matrix?
Mutation.
This will tell you what is a mutation rate, right. Actually, we can derive the scoring matrix in various aspects. So, for example, if you take the nucleic acids, you can see the purines and the pyrimidines, right. You can use transversions and transitions, right. Based on that, we give penalties. If you discuss about the case of amino acids, you can do the physical chemical properties, right, and the genetic code, right, how many variations, right. Then finally, you can do it with the actual rates, the actual mutation rate. For example, if you take the set of homologous sequences, say about 80% or 90% sequences, okay. In this case there will be several mutations, and see what is the most probable mutations. Using these probable mutations, you can derive the matrix, right, from one amino acid to other amino acid. So, if you remember, you can see there are some cases they are acceptable, some cases it is not acceptable. Mainly if you mutate same residues, there are no mutations, that is highly preferred ones. Even in that case, depending on the amino acids, for example, compare the tryptophan or the cysteine as with alanine, tryptophan is more conserved than alanine, right. Likewise, if we have similar type of amino acids, for example, alanine to valine, right. So, this is preferable or not? Is preferable, right. For example, if alanine to lysine, it is not preferable, right, either the physicochemical properties or the scoring matrices, right. Scoring matrix you can develop from the mutation rates of amino acid residues, how far each residue is mutated to different types of residues, right, we can make that. So, this is how we made the scoring matrices, right. What are different matrices we discussed?
PAM and blossom.
PAM matrix and the blossom matrix. What is PAM matrix?
Percent accepted mutation.
Position accepted mutation matrix, right. So, the how to derive the PAM matrix, what are the various aspects we need to consider to derive this PAM matrix. So, first we say the frequency of these amino acids, length of the sequences, and how this residue is mutated and which residue is mutated to which residue, right. For example, if it is Aij, right. So, how many times alanine is mutated to valine, right. How many times alanine is mutated to all the other residues, 20 residues, how many alanine is present in the sequence, right, what is the length of the sequence. So, all these aspects we need to consider for developing this PAM matrix, right. Right. Then what is blossom matrix? This blocks substitution matrix, right. So, what is the difference between PAM and blossom?
PAM is from homologous sequence.
Yeah, this takes almost all these alignments to see the mutation rates. Blossom will take these conserved regions. So, they can see to avoid these gaps and then see the conserved regions how they can make this alignment. Then we discuss dynamic programming. What is dynamic programming?
Divide the problem into
Yeah, divide the problem into small pieces, finally solve the problems and then finally combine together and solve the whole system, right. So, that is called dynamic programming. So, what are the different aspects? So, we use dynamic programming for the sequence alignment. What are different two different alignments? Global alignment and the local alignment, because we can have two sequences, you can use various ways to align these sequences, right. So, there are different pathways, right. So, we use two different alignments. What is global alignment? Whole sequence, right? Whole sequences. What is local alignment? Right, only the small part of the sequence we can align, right. What is the algorithm we use for global alignment? Wundt-Stiedelmann algorithm, right, Wundt-Stiedelmann algorithm. What are the conditions used to fill the matrix in Wundt-Stiedelmann algorithm?
Match,
Maximum of
match.
This is the match or mismatch. Right, match we have score, mismatch we have score,
Gap.
then gap, that is insertion or deletion, right. So, we have the rank, we have the score for each case, we have match score, we have mismatch score, we have insertion gap penalty for insertion deletions. So, based on this you can give the maximum value provided from all these three values, right. So, what is local alignment? What is which algorithm we use for local alignment? Smith-Waterman, right. So, what is the condition for the Smith-Waterman algorithm?
Match, mismatch and 0.
This is plus 0. So, in this case there is no negative values, right. In this case you can restrict for the small gaps, any small matches, fine. So, how to align these sequences? If we have two sequences, right, first we see the very different options, right. For local element, see the 4 values, then fill the value numbers, and from the back see the highest score, you go back to trace back, then accordingly you can put the gaps, right, and then you can make this alignment, right. Then what are the various programs we use for sequence alignment? BLAST is widely used tool. What is expansion for BLAST? Basic local element search tool, right. So, what are the various features of the BLAST?
Can do
Right, you can search for any sequences, you can get all the sequences which are matching with the particular sequence and align the sequences, right. So, you can what are the various features we get, what are the various scores we get from BLAST? right. We can see the p values, we can see the e values and the similarity,
Max score.
right, maximum score, right, identity, these scores we can get in the BLAST, and coverage, right. From that you can see whether a sequence alignment is perfect or not. For example, this is a sequence. So, in this case what is the sequence identity? So, how many identical sequences?
3 positions.
If you take this one, right, then you can see the coverage of 6 for 8, right. So, this is the match, here is the match, here is the match. So, 3, right, 3 for 6 you can see, right, and the similarity, okay, this is similar, T and A similar. So, you can take this, you can see 4 by 6, right, because the coverage is only 6, right. You can see this, the coverage is only 6 by 8. 6 by 8 means how many percentage? 70 percentage? Yeah, 70 percentage, right. And then what is the multiple sequence alignment? Align more than 2 sequences, right. So, you can this case there are various ways to get the multiple sequence alignment, right. So, you can first see the similar sequences, then go with these distant related sequences, they put it together and you can see which residues, right, which come in the same position, right. You can get the information from multiple sequence alignment. What are the online resources available for multiple sequence alignment? Cluster omega,
Mapped.
then mapped, muscle,
Mass.
right. So, you can use various software, right, for the aligning the sequences, these multiple sequence alignment. Then we discussed about the conservation. What is conservation?
This
Yeah, a residue is occupying the same position, right. If you have the 100 sequences, among the 100 sequences take position number 5, it is alanine. How many sequences they have alanine at position number 5? So, this will give you the information regarding the same amino acids presents at the same position, right. So, how to calculate the conservation score? There are 2 step procedure. First, what you have to do?
frequency
First, we have to get the frequency of amino acids and then
score.
we need to convert it into score, right. What are the various ways to get the frequency of amino acids?
Weighted frequency.
Unweighted frequency and the random, right, the independent counts. How to get unweighted frequency? How to get this frequency?
Count of the
N of I by N, right. So, for each position, that how many times each amino acid is present, right, and then see how many amino acids at a particular position. Then we get the frequency, then we convert into score. How to convert the score? What are the various ways to convert the frequency to score?
entropy based.
Right, entropy based, variant based and sum of pairs, right. You can use the different ways to get this, right. So, what is the program available to get the conservation? AL2CO, right. How it works?
We have to give multiple sequences.
Yeah, we can give the multiple sequence, well, this is alignment, we give the multiple sequence alignment, then they use any of these algorithms. So, if what we can choose whether you need entropy based measure or the variant based measure, right, and also the frequency also you can choose, based on that you will calculate the score. Then you normalize the score, right. What is the formula to normalize the score? X minus x bar by sigma. So, you can get kind of G score, you can normalize the values. Then what are other analytical resources to get the conservation? A conserve, right. So, you can here if you give the PDB-ID, automatically it will get the sequence and collect the homologous sequences and get this conservation, right, for each positions. This will give you a pictorial representation. They see the highly conserved as well as the less conserved, right, variable regions we can see. It can also provide the information regarding each position, right, what are the other residues, right, which are present in the particular position if any residue is the variable. So, then we did the phylogenetic trees. How to construct the phylogenetic tree? What the information we get? How the sequence related, which 2 sequences are closely related, which are the distant related, right. So, what are the various ways to construct the trees? UPGMA method. This is the very popular common method. So, what is the basic information required to construct the UPGMA method? The tree distance, right? See the mismatches. Depending upon the mismatches, we can say, right. If two sequences are less mismatches, that means they are closely related, right. So, you can construct this phylogenetic tree. Then we discussed about the phylip, right. So, this widely used program. So, you can use the phylip, right, to construct the phylogenetic trees. Okay, then see, you can do this from amino acid sequences. You can get the alignment, you can get the multiple sequence alignment and the conservation close related sequences, all the information you get from the sequence.